The topic that I am going to explain today is nucleic acids and protein synthesis. These are some of the major highlights, the important points that we'll try to understand. First of all, obviously we need to understand what basically the nucleic acids are, their location inside an organism, their structure and morphology, and how many different types of nucleic acids are there. Then we'll try to understand the difference between the DNA and RNA. And once we have built the basic concept and understanding, we look into the details of an important process of biology that is also called the central dogma of life. It basically includes two steps, transcription and translation. All of these concepts will be elaborated in the further slides. Let's begin with an introduction of nucleic acids. So the nucleic acids are naturally occurring chemical compounds. They are not synthetic in nature, they are already present inside the cells of different organisms. They are high molecular weight polymers. What do we mean by the polymers? Polymers are those substances that are made up of smaller subunits or monomers. Likewise, nucleic acids are also polymer in nature. This means that they also have their smaller subunits, which are called nucleotides. These nucleotides act as the building blocks and are joined together to different types of bonds to make the nucleic acid. If we talk about the types of nucleic acids, then these are DNA and RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. DNA, as we already know, is present inside the nuclei of the cell. Here we can also see in the picture a human body is represented. All the human beings and all the complex organisms are multicellular. This means that they have a lot of different types of cells that in turn make up the human body. So the nucleus of the cell contains a very well organized structure which is called chromosome, which has the DNA molecule wrapped over it. The DNA then contains the small segments which are called genes. These are basically the functionally active chunks of DNA that encode for various traits and attributes of an organism. For example, height, eye color, hair color, and expression of several different diseases is controlled by the expression of these segments of DNA which we call genes. Here I have used a term functionally active chunks of DNA. What do we mean by this? Functionally active chunks means that all the information that is contained inside the DNA is not needed. It is not functionally active. We only consider those chunks of sequences, those segments of DNA that are functionally active, that are transcribed and translated into some protein. And that protein is basically the controller of expression of different traits. In short, the DNA basically controls all the genetic information that is required for the proper functioning of an organism. Not only that, it is also responsible for the transmission of this genetic information to the next generation through inheritance. The other type is RNA. RNA basically is not present all the time inside the cell. It is synthesized only when it is needed. It is synthesized. It is basically a copy of DNA. It works in synchronization with DNA to synthesize the proteins. So whenever a protein is needed inside the human body or inside the cell, only then RNA will be synthesized. Otherwise, it is not already present inside the cell. Now let's discuss nucleic acids in terms of structure and morphology. Here we are discussing the building blocks of DNA and RNA. As I said before, that both DNA and RNA are polymers that are made up of small subunits called the nucleotides. All the nucleotides have the same structure, same basic structure having only three components, which are a phosphate group, a pentose 5-carbon sugar, and a nitrogenous base. In this picture, we can see a 5-carbon sugar is present. At carbon number 1, we have a nitrogenous base attached to it, and at carbon number 5, we have a phosphate group attached to it. All the nucleotides are going to have the similar structure, a 5-carbon sugar, and carbon number 1 is going to have any nitrogenous base, and carbon number 5 is going to have a phosphate group. This is similar for all types of nucleotides. This means that all the nucleotides have the same backbone. How the difference arises is basically dependent on the type of the nitrogenous base that is attached to the first carbon of the pentose sugar. There are different types of bases we are going to discuss in the next slide. Okay, so here we have the structural representations of all three components of the nucleotides. The first component is phosphate group. The next one is a pentose 5-carbon sugar, and the third component that makes the nucleotides is a base, a nitrogenous base. So I said that all the nucleotides have the similar basic structure. A 5-carbon sugar that has a base attached to the carbon number 1 and a phosphate group attached to the carbon number 5 of it. The difference arises only when we have the different types of bases attached. So there are two different classes of bases. We classify the bases in two different categories. These are purines and pyrimidines. How are they different from one another? Purines have the two ringed structures that are attached to each other. For example, in purines we have adenine and guanine. And in the picture we can see that they have two ring-like structures that are attached to each other. And pyrimidines are the single ringed structures. This means that they have only one ring structure. There is no another ring that is attached to it. And the pyrimidines are thymine, uracil, and cytosine. And whenever we have to represent a nucleotide, we represent them only by writing the first letter of the base. For example, if we have the nucleotide that contains a base adenine, we will write A in its place. To represent that nucleotide, we are going to only write A in capital letters. If a nucleotide has G attached to the place of the base, guanine, then we will write G. If it has thymine, then we will write T. If it has uracil, then we will write U. If it has cytosine, and then we will write C, all in capitals. This is because the phosphate group and sugar remains the same for all nucleotides, only the base differs. So when the difference arises just because of the base, we only represent the nucleotide with the name of the base in simple words. Now let's discuss the differences between the DNA and RNA. As we have discussed that there are different types of bases that make the nucleotides different from one another. These bases are thymine, cytosine, guanine, adenine, and uracil. In case of DNA, four bases are present: thymine, cytosine, guanine, and adenine. These four bases are specific to DNA. While in case of RNA, we have cytosine, guanine, adenine, and uracil. Uracil is not present in DNA. In its place, we have thymine in DNA. While on the other hand, in RNA, uracil is present instead of thymine. So this is how they differ in bases. Now, if we morphologically consider DNA and RNA, then DNA has a double helical structure. It is double-stranded in nature, while on the other hand, RNA is single-stranded. What do we mean by this? This means that DNA has two different strands that are bonded together through complementary base pairing. This means these different color strands are shown that are joining the two double helices of DNA. The colored strands represent the different bases. Pink strand shows thymine, green strand shows guanine, blue strand here shows cytosine, and orange strand here shows adenine. And what is complementary base pairing? This means that these bases are very specific in nature. Thymine will only make a bond with adenine, and adenine will only make a bond with thymine. Similarly, cytosine will make a bond with guanine, and guanine will make a bond with cytosine. It cannot happen that thymine makes a bond with guanine and cytosine makes a bond with adenine. It is not possible. This is the selective nature of DNA, and through this complementary base pairing, the double helical structure of DNA is maintained. While on the other hand, RNA has no double helical structure, so it does not require any complementary base pairing. Because to maintain the double helical structure, we need something that can bind the two helices together. So that binding is brought about by the hydrogen bonds between different types of bases. There are hydrogen bonds between thymine and adenine, and hydrogen bonds between cytosine and guanine that are responsible to maintain the double helical structure of DNA. While in case of RNA, there is a single strand, there is no need of complementary base pairing. We only have the single strand and a base, the bases are attached to it. Which bases? Only C, G, A, and U. Here I am using only the first letters, as I said earlier, that nucleotides differ from one another on the basis of their nitrogenous bases. So we can just represent the nucleotides with the help of the bases. The other difference that arises is due to the pentose sugar that is present in DNA and RNA. DNA, as the name indicates, it contains deoxyribose. Deoxyribose. While on the other hand, RNA contains ribose sugar. How are they different from one another? Here we can see the structural representation of a 5-carbon sugar. At second carbon, in case of deoxyribose, DNA, in case of DNA, the second carbon only has a hydrogen group attached to it, while on the other hand, in ribose sugar, the second carbon has a hydroxyl group attached to it. Oxygen is present here, so it is oxyribose, which in generally is known as ribose sugar. But in the case of DNA, oxygen is removed. There is no hydroxyl group on the carbon number 2 as it is present in the carbon number 2 of RNA. There is no hydroxyl group in DNA at carbon number 2 of pentose sugar. That is why we call deoxy. This means that the oxygen is removed from here. The oxygen is not there, the hydroxyl group is not here, only the hydrogen group is present at the carbon number 2. That is why it is called the deoxyribose sugar, which in turn is present in the name deoxyribonucleic acid. So these were the three basic differences. One is the morphological difference, the other two are the chemical differences. The morphologically or structurally, DNA is a double-stranded structure, RNA is a single-stranded structure. DNA contains deoxyribose because oxygen is removed from the carbon number 2 of pentose sugar, while RNA contains a ribose sugar where we have a hydroxyl group present at the carbon number 2 of the pentose sugar. The third difference is we have 4 bases: thymine, cytosine, guanine, and adenine for DNA. And we have 4 bases: cytosine, guanine, adenine, and uracil for RNA. Thymine is present in case of DNA, and in its place we have uracil in the case of RNA. So these were the basic differences between DNA and RNA. As we have already learned about the DNA, RNA, their differences and their functions inside the cells, now we can move to a bit complex process that is called the central dogma of life. All the traits and all the attributes of humans, plants, and different complex organisms are controlled by the expression of the functionally active chunks or genes that are present on the DNA. When those genes are expressed, what do we mean by the expression? This means that those genes are transcribed in the form of RNA, and then those genes will be translated into the form of the protein. Those proteins in turn will control an expression of an attribute. For example, different proteins are controlling different metabolic reactions. The eye color, the hair color, intelligence, weight, height, the expression of different diseases. For example, thalassemia, sickle cell anemia, diabetes type 2. These are all controlled by the genes and in turn the protein products of those genes. So, how the central dogma of life works is, first, the DNA basically contains all the information, which are basically the functionally active chunks, as we have discussed earlier, the genes. That information will be decoded and copied in the form of single-stranded RNA, and that RNA will be responsible to synthesize the protein according to the information it has gained from DNA. This whole process is called the central dogma of life. It has 2 steps: transcription and translation. Transcription is a procedure in which the information from the DNA is copied in the form of RNA, and translation is a procedure in which the information from RNA is ultimately converted into the protein product, and that protein works and controls and measures all the attributes of the organisms. Okay, so now we will discuss the 2 steps of central dogma of life: transcription and translation. As we have already learned that DNA is the hub of genetic information. Everything that happens inside the human body is not only the human body, we are generally talking about the multicellular organisms and even the unicellular ones that contain the DNA inside their cells. Everything that is happening inside the body of an organism is controlled by the genetic information that is present in its DNA. How is it controlled? By controlling, we means that whenever a certain process is to be carried out inside the cell, it will take instructions from DNA. So in the first step of carrying out those instructions, we need to copy the genetic information from DNA into a functional strand which is called a RNA. Now RNA has different types. Three basic types of RNA are messenger RNA, transfer RNA, and ribosomal RNA. In this case, in the case of transcription, we will be discussing the messenger RNA. As the name indicates, messenger. This means that it has to carry a message from somewhere to someone else. For example, In the case of transcription, messenger RNA molecule will carry the information from DNA strand to the ribosome. So that is why it is called the messenger RNA. It carries the message, it carries the genetic information. The genetic code that is present on DNA will be decoded in messenger RNA, and then messenger RNA will continue the process to ultimately synthesize a protein product. Now, as far as the process of transcription is concerned, it has three steps. The first step is initiation. The second step is elongation. The third step is termination. If we consider the diagram here, we can see we have a DNA double helical structure and an enzyme which is called RNA polymerase. All the complex biochemical reactions take place through the help of different types of enzymes. In the process of transcription, the enzyme that is helping is called RNA polymerase. Now we have a green colored region which is called promoter region. Promoter region basically is a region that contains the codon. Codon for initiation of transcription. It is basically a signal point that tells RNA polymerase that you have to start transcription from this point. This is the beginning point of the transcription, which is called the promoter region. So RNA polymerase binds to the promoter region, and what happens next? The basic work it has to perform is to unwind the strand of double-stranded structure of DNA. Only then the transcription will be able to happen. Why? Because if it is intact, if it has complementary base pairing, those bases have different numbers of bonds between them. How is it going to copy the information from DNA to RNA? So first step is to identify the initiation region, which is called promoter sequence or promoter region. It will have a codon, for example, AUG or ATG in case of DNA. ATG in case of DNA stands, this is the codon for the amino acid methionine. Methionine is an amino acid that is present at the first number for all the proteins. No matter which protein is it, it will always have methionine in the first position. So most of the promoter regions start with A, T, G. In case of DNA, we are saying T because thymine is present here. As I said earlier, we can only represent the nucleotides using the first letters of the names of bases. So we are representing gene, a protein here. One basically protein is encoded by three triplet codon of nucleotides. A, T, G encodes for a one amino acid. So one amino acid is encoded by the triplet codon, three nucleotides together. Then A, C, G, A, C, C, G, T, C. These are the different codons for other different amino acids, maybe cytosine, maybe histidine, lysine, etc. So for promoter region, we always have a constant amino acid which is methionine ATG. Okay, so RNA polymerase first identifies the promoter region. After the identification, it binds to the promoter region. And when it binds to the promoter region, next step is to unwind the double helical structure. It will break the bonds, the complementary base pairing, the bonds between the bases of the DNA. The double helical structure will open. Now, the process of elongation starts. Next step, second step, elongation. RNA polymerase will continue reading the triplet codons. First was ATG, next was CCC, Third one was GTA. Next was some other codon. Then the process continues and it carries on reading the sequence until the termination point reaches the third step of termination, which is the stop sequence. DNA double helical structure. DNA carries the information from the starting point as well as the ending point of transcription. As the RNA polymerase identified the promoter regions to start with the transcription, it also has to identify the termination region or stop sequence to end the process of transcription. This is how, when it reaches the stop sequence in the termination process, it stops copying the information of DNA in the form of messenger RNA. So, how it was copied? The first codon was ATG. On the messenger RNA, it must have been A, U, A, C. Fine. The complementary base pairing. A binds with U, G binds with C, C binds with G, U binds with A. Likewise. So the messenger RNA that is synthesized inside the nucleus will be transported into the cytoplasm through the nuclear pore of the nuclear membrane. The basic information or the genetic information has been copied in the form of messenger RNA. Now the process of translation begins. The transcript or the copy of DNA has already been synthesized in the form of messenger RNA. Transcription is over. That copy of DNA in the form of messenger RNA was transported from nucleus to the cytoplasm. In cytoplasm, we have the ribosomes. These are the protein factories, and transfer RNAs and different types of amino acids are already present inside the cytoplasm. As we discussed that there are three different types of RNA. Messenger RNA was used in transcription. Now the other two types, these are transfer RNA and ribosomal RNA, will be used in translation. Ribosomal RNA is basically the structural constituent of ribosome. Ribosome as a whole will participate in the process of translation. And transfer RNA, as the name indicates, it has the function of transferring the amino acid from one place to another. Now, if we just consider the diagram, we have a single-stranded messenger RNA that carries the codes, that carries the genetic information from the DNA. The main code was on the DNA. It was copied in the form of messenger RNA. See, we have CUC, UUGGG, UCC. These are all the codons for different types of amino acid. For example, CUC will encode for one amino acid. UUG will encode for another amino acid. GGU will encode for another amino acid. As I explained earlier, that one amino acid is encoded by the triplet codon. Triplet codon. Three nucleotides or three bases together form one codon for one amino acid. When the translation begins, what happens is messenger RNA comes out of the nucleus into the cytoplasm. Now, the ribosome binds. A ribosome has two different subunits, larger subunits and smaller subunit. Ribosome binds with the messenger RNA using its smaller and larger subunits. Now, the ribosome then starts reading the sequence. Ribosome then starts moving triplet by triplet, not one by one. C itself encodes for nothing. U itself encodes for nothing. Only the triplet codon will make sense to transfer RNA and ribosome, and only then they will be able to transfer one amino acid or the other amino acid from the vicinity of the cytoplasm. So, transfer RNA will bind with these triplet codons and they will read those triplet codons one by one, triplet by triplet, I mean. CUC, first of all, the transfer RNA reads the sequence, the codon CUC. It says it is encoding for glycine amino acid. It will find glycine from the vicinity of the cytoplasm and it will transport that glycine to the near the messenger RNA. Next code UUG. The next RNA, the next transfer RNA will come. One transfer RNA can bring only one amino acid. The transfer RNA that is designed to bring glycine will only bring glycine to the place where the protein is being synthesized. It cannot bring leucine or tyrosine or serine or alanine or valine or any other amino acid. So these are also specific in nature. When one codon is over, what will happen? The next transfer RNA will come. It will read the second codon UUG. It identifies that it encodes for serine. Now it has to find serine and what will happen? We already had glycine from the first codon. Now serine comes. These are the two amino acids. What will happen? The peptide bonding will take place between the two, because we know that proteins are also the polymers. Proteins are also polymeric in nature. They are made up of amino acids. Amino acids are joined together through peptide bonds. When the huge number of different amino acids are joined together through peptide bonds, a multimeric or a polymeric structure of a protein is synthesized at the end, which is functionally active. So only one amino acid or two amino acid, they cannot perform any function. To make a functionally active protein, we need to join these amino acids, these subunits together through peptide bonding. So first amino acid was glycine, the other was, for example, leucine. It was joined together. The next code is GGU. For example, the next transfer RNA reads the code GGU. It says it encodes for tyrosine. It will bring tyrosine from the vicinity of the cytoplasm, and the tyrosine will be joined to the previous two amino acids through peptide bonding. Now we have the chain of three amino acids, glycine, leucine, tyrosine. All of them are joined together through peptide bonding, and the chain continues to grow until we are left with the functionally active protein unit that is polymeric in nature. This means that it has hundreds or thousands of amino acids that were joined together through the peptide bonding to give us a functionally active required protein. Okay, so this is the whole process of translation that takes place using messenger RNA, ribosomes, and transfer RNAs. So now I am going to summarize whatever we have learned. One thing I would like to mention is whatever we have discussed is as per the syllabus of Cambridge International A-Levels Biology Book 9700. So we discussed about nucleic acids and we learned that these are also the polymers. They are made up of the smaller subunits which are called the nucleotides, and the nucleic acids are of two types, DNA and RNA. DNA is present inside the nucleus of the cell on the chromosomes, and RNA is not already present inside the cell. It is synthesized only when it is needed. When a cell has to synthesize a protein, only then the messenger RNA is synthesized. So nucleic acids are the polymers of nucleotides. All the nucleotides have the same basic structure comprising of three components, which are the pentose, sugar, the phosphate group, and the nitrogenous base. All the nucleotides are same except for the base they contain. So there are different types of bases, and those bases are classified as purines and pyrimidines, and this classification is basically based on the structure they have. Some of the bases have the adenine and guanine have the double ringed structures, while the other bases have the single ringed structure. So structurally they are classified into the two categories, purines and pyrimidines. Then we also learned that there are some differences between the DNA and RNA. DNA is a double helical structure. Double helical structure of the DNA is maintained with the help of the complementary base pairing. This means that the two bases are bonded together through hydrogen bonds. Two bases, in a sense, that adenine makes bond with thymine, cytosine makes bond with guanine in case of DNA. And in case of RNA, there is no double helical structure, so complementary base pairing is not needed. And RNA is a single-stranded structure. DNA contains four different bases, which are adenine, thymine, guanine, and cytosine, A, T, C, and G. And RNA contains cytosine, guanine, adenine, and uracil. It does not contain the thymine. So it contains C, G, U, and A. Okay, so the amino acids encode for these three triplets of codons together encode for one amino acid. Fine. A, U, G, G, C, U, G, G, C. These are few examples of the triplet codons that encode for the different amino acids. For example, lysine, histidine, cysteine, etc. The other difference is the presence of deoxyribose and ribose sugar. DNA contains the deoxyribose. This means that it does not contain the hydroxyl group at the carbon number 2 of pentose sugar. Rather, it only contains the hydrogen group attached to it. While on the other hand, in RNA, we have the hydroxyl group. Oxygen is already there. Hydroxyl group is attached to the carbon number 2 of the pentose sugar. That is why we call it a ribose sugar, which in turn is indicated in the name, ribonucleic acid. So these were the three basic differences between the DNA and RNA. Then we discussed what is the process of the central dogma of life. That whenever a process takes place inside a cell, first of all, we need to take the information from the DNA, which in turn is decoded in the form of messenger RNA, and messenger RNA then helps in the synthesis of the protein, and the protein regulates and controls the expression of various traits. In central dogma of life, there are two steps, transcription and translation. First of all, the DNA, that is the hub of genetic information. The information from the DNA is encoded in the form of messenger RNA. It happens in the three different stages. First is initiation, next is elongation, and the third and final one is termination. In initiation, the RNA polymerase enzyme identifies the region, the promoter region, where it has to start the transcription. Then it binds itself to the promoter region. Then it unwinds the double helical structure of DNA and start copying the information. The strand of DNA that is being copied is called the sense strand. Sense strand. Only the information on the sense strand is copied in the form of the messenger RNA. And in the step of elongation, the RNA polymerase continues reading the sequences, the codons from the DNA, and continues converting those codons into the mRNA sequence until it reaches the termination point, which is the termination sequence. And in the final step of termination, the RNA polymerase detaches itself from the DNA, and messenger RNA single-stranded structure is liberated, and then it is released from nucleus to the cytoplasm. Then followed by the transcription happens the process of translation. In translation, the messenger RNA that is now inside the cytoplasm is carried to the ribosomes. Ribosomes are the factories of protein synthesis that contain the smaller and larger subunits. Those two subunits join with the messenger RNA, and here few other factors take part. For example, transfer RNA and amino acids that are already present inside the cytoplasm. So the ribosome and transfer RNA continue reading the sequences from the messenger RNA, the codons, and continue joining the amino acids based on the information of the codons. Those amino acids are joined and linked together through peptide linkages. And as a result, we get the final product, which is a protein. That protein controls the expression of all the traits inside the organism.